Good afternoon, evening, morning, wherever you are. We are delighted to have you join us for this 10th annual Frank and Miller lectureship, which tonight is going to be a panel instead, as you will see, which is our SMHS, our GW School of Medicine and Health Sciences bicentennial signature event this year. We're delighted to have you here with us. It really is a celebratory moment for us here at GW as we begin to be fully formed and back on campus here. My name is Dr. Barbara Bass. I'm the Dean of the School of Medicine and Health Sciences here at GW and the VP for Health Affairs. I also happen to be the CEO of the GW MFA, our faculty practice plan. So it's great to have representatives of all of those different domains here with us tonight to talk about this important topic of translational medicine, translational science in our campus here, as well as more broadly. It's really great to welcome people here for this alumni weekend. As you know, last year we were all virtual. People are beginning to trickle in now to be here physically on campus, which is thrilling. I've just visited the mall to see the grand preparations for the long deferred recognition of last spring's commencement events. It's quite exciting here at Foggy Bottom right now. And I hope that those of you who are joining us here for the celebratory events enjoy your time on this beautiful fall few days here in Foggy Bottom. And as you restore those memories of your time here as a student or at the GW School of Medicine and Health Sciences. This is a particularly important alumni event, a weekend for us. Not only is it our 197th alumni celebration, or at least celebration of previous graduations. We haven't actually been doing alumni events for that long, but we've been graduating students for 100. Has been graduating students, enrolled its first student 200 years ago. So this is our GW bicentennial of the university with our own School of Medicine and Health Sciences event to follow in 3 short years. So it's quite celebratory around here this moment. I will say that I've been here for about 18 months now. I arrived 2 months before the pandemic began, and I think just now we're kind of beginning to creep back into something that feels a little bit more normal. But as we look back on this last 16 months of this ongoing, not quite over yet, pandemic, I think more than anything in my lifetime in academic medicine, this period of time has demonstrated the incredible value of academic healthcare centers. When you look at what we have done over this period of time across the nation, here at GW, but also across the nation, ranging from discovery science to clinical trials, to rapid implementation of new clinical protocols, all in the face of this dread, novel, deadly coronavirus, and of course building from scratch a new set of highly effective and safe vaccines. Only academic medicine healthcare centers have that built-in capacity from discovery science, clinical trials, clinical implementation, distribution in our nation to do that kind of vital work. Yes, we partner with industry. Yes, we partner with other groups, but really that is our core mission as an academic medical enterprise. And so I'm so proud of the work that our teams here at GW have done over this time. And yes, we've done all of that, from discovery science relative to this novel virus, to participation in the vaccine trials, the Moderna trial here, to rapid implementation of best care practices in our ICUs on the front line of our healthcare providers, as well as just seeing the rapid implementation and distribution of public health measures through the abundant activities of our students, our faculty, our staff, to protect the health and welfare of our Washington, D.C. community. It's been an exceptional and wonderful journey, and I'm really grateful to all of those. So we've lived translational medicine in its fullest form over this last 18 months. But now, as we're coming out of this, it really is kind of nice to realize, it's more than just COVID here. You know, we actually have a lot of really important work to do on so many fronts in healthcare. And the way that we distinguish ourselves as at GW and as academic healthcare research institutions overall, is we do that complete portfolio of scientific discovery, trials, and implementation. We talk a lot about sort of bench to bedside. What does that mean? Well, it means we do that full scope. And tonight we have with us representatives of each phase of that bench to bedside process, that discovering why, from molecular and genetic mechanisms of disease and viruses and molecules and organisms to developing those targeted therapies that will allow us to develop new therapies or diagnostics to improve the care of our patients, to the clinical trials that demonstrate the value of that discovery science and effectiveness of that. And then, of course, to implementation and even commercialization. It's that spectrum that represents translational science and what we're going to talk about tonight. So I'm really proud of the work that our scientific and healthcare teams do here at GW. And I think you'll see that we have really an abundance of riches in terms of our depth of expertise and that complex and marvelous process of translational science. Again, all of it driven with the goal of improving the lives of the patients that we take care of. So we're going to start this with a really nice, I think, a really nice example. How does this all work? And again, we keep the patient at the center of the focus of everything we do, from basic discovery to treatment. And we are really fortunate to have with us tonight my video, previously made a video, a patient of Dr. Henry Kaminsky, who's here with us tonight, our chair of neurology and really an internationally acclaimed investigator from bench to bedside, from basic science to implementation in the field of myasthenia gravis. He has one of his patients with him tonight, and thank you to Ann Banner for having put together this beautiful video. But the patient's name is Dr. Rosia Paula Caballero-Gil. She's a paleontologist, a paleooceanographer. She also has myasthenia gravis, and she's a patient who's been treated by Dr. Kaminsky on one of his clinical trials over the last couple of years. She tells this story beautifully, and I hope that you will all enjoy seeing this vivid example of why we do the research we do.
Hi, my name is Rocio Paola Caballero-Gil. I am a paleoclimatologist and paleoceanographer. I'm really a geologist, and I'm Peruvian born and raised. I have 2 children. I'm married to the love of my life, and I live with a big family in my home, so it's a multicultural, multi-generational house. For me, everything happened in stages. It was everything in the year of 2011. April of that year, I was doing Zumba, and I was a little out of shape, and I just collapsed to the floor. I plummeted. And I thought it was because I was out of shape. And so I got up and I was shaking because I fell, and I've never done that. And then I did it again. I was like, okay, I'm not that clumsy, this is something else. And then by the time I went back home in November, I couldn't chew a steak that I was eating with my husband. So I went to the doctor. The neurologist immediately did a strength test and decided it was myasthenia.
Myasthenia gravis, first of all, is an autoimmune disease. It affects critical aspects of nerve-muscle communication. So then all the symptoms come from that. Patients can have really visually disabling double vision, droopy eyelids to the point they can't see, speaking problems, trouble swallowing, breathing problems, and in the most severe cases, they need to be on a breathing machine. So it can be just really a life-devastating disease.
I got myasthenia when I was an adult. I think it took me years to really come to grips and understand my new person. I had to mourn my scientist in my head and where I was going and what I was going to do and the family I wanted to have, and mourn that and then be adjusted. So it was a long process to really come to grips with who I was.
I met Rocio in 2018, And she had come to me already with myasthenia diagnosed back in 2011. She came to me and she was failing prednisone therapy. She'd already had a surgery called a thymectomy. She was getting intermittent dosings of intravenous immunoglobulin. She was on a common medication, pyridostigmine, and she still really couldn't do her job. She had profound difficulty taking care of her children and her husband, and really was essentially housebound from my perspective.
Dr. Kaminsky had talked about the research, so I said, you know, if nothing is working and I need something to work right now, maybe the research route is an option. So I got in touch with Dr. Kaminsky and I said, you know, out of desperation, let's do it.
When she entered the trial, within the first weeks, months, she already was improving. So it was really a dramatic effect for her. And then over the course of the study, she's continued to do very well.
I felt the progress like skyrocket. It was like day and night. And with this drug, you know, the improvement was fast. And the ability came back fast. And so I was like, wait a minute, I can actually read a book to my children at night without having to stop after every page. Or I can go up the stairs and play with my daughter. And so those were things that I was already used to, not being able to be the mom that I wanted to be, the scientist that I wanted to be. And so it took a lot. But it was good, you know, and of course it wasn't 100% great. I still couldn't drive, I still can, but at the same time I was hopeful that maybe someday I can still try.
I think the most important aspect that sometimes gets lost is the critical nature of the patients. You know, they're putting their lives, quite frankly, on the line. There's no guarantees when you're in a clinical trial that a complication's not going to occur. And so I think it's an incredibly scary and brave decision to enter into a clinical trial. But ultimately, it's just so humbling for them to enter into this and take a risk and, you know, potentially lead to a treatment that is accepted across the world and improves people's lives. So it's really the patients who are doing all the work.
Thinking of the value of being part of a clinical trial, I feel that the value is immense. It's so different from what I was used to. My symptoms have improved dramatically. They're not gone. I'm still taking my meds, and I still have to go and take the scary meds that I didn't want at some point. But I still feel the difference.
So here at GW, we're actually doing this work as part of an NIH grant to understand myasthenia and just rare diseases in general. My goal, quite frankly, is to get a cure. You know, otherwise, why do things? But to get to that cure, there's a lot of different steps, right? There's the basic scientist still understanding the disease better, and as it's like peeling an onion, you understand it, then you see that you don't understand this point. I'm hopeful that we can actually get to a cure, which is what everybody wants.
Through this journey, I've been lucky to have the support of my family, my husband, my children, and Dr. Kaminsky was there for me, and I trusted him enough to say, yeah, I'm just gonna do this. I just wanted to say thank you to all of them, because this wouldn't be what it is without them.
Well, we are really so grateful to Dr. Caballero-Gil for sharing her story with us, and of course to Dr. Kaminsky as well for being part of that really informative, raises many questions about what we can talk about tonight in terms of this process of translating basic science into clinical application to cure disease. All right, let's move on to our panel now, because we have really a terrific group. And interestingly enough, they all span sort of different parts of that rainbow, that scope of translational science, from discovery science through to clinical implementations and the barriers thereto. So let me briefly introduce our speakers. We have Dr. Rong Li, who is the Chair and Ross Professor of Basic Science Research in the Department of Biochemistry and Molecular Medicine at the School of Medicine and Health Sciences. And his research is focused on breast cancer and the role of BRCA1 in tissue-specific tumor suppression. We're going to hear just a thumbnail sketch from each of our speakers about their research. Secondly, we have Dr. Antonia Sepulveda moving along from that basic discovery to that next phase of translational science. Dr. Sepulveda is the professor and chair of the School of Medicine Health Sciences Department of Pathology. And her research work is focused on the genetics of gastrointestinal malignancy. And she'll tell us a little bit about how that falls into this translational spectrum. As you see, we also have Dr. Henry Kaminsky, who we just met in our video, who is the chair of our Department of Neurology and the Meta Amalia Newman Professor of Neurology here at the School of Medicine and Health Sciences. And as you see, he has a long career dedicated to solving this condition of myasthenia from basic discovery science to clinical treatments. Next, we have Dr. Mandy Pratt-Chapman, who is the Associate Center Director for Patient-Centered Initiatives and Health Equity at the George Washington Cancer Center. She's done a lot of work on evidence-based cancer survivorship and patient navigation practices. She's also an advocate for ensuring equity in patient-centered care with a focus on many underrepresented groups. And she will bring that perspective of you got to have access to the system to be part of the innovation and part of the improvements along the way. So she'll talk to us a little bit about that. And last but by no means least, we have Dr. Keith Melanson, who is our head of our professor of surgery, as well as the head of our transplantation program here at George Washington University. He specializes in kidney transplantation and has really been a vital innovator in the field of the chains of transplantations to facilitate optimal use of organ resources, as well as clinical trials relative to immunosuppression. And he's going to talk to us today mainly about his work in decreasing barriers to translational science and implementation strategies in the Black community relative to kidney transplantation, where he has been a nationally recognized leader in effective delivery of that goal. So with those brief introductions, I hope that we could have each of you give us a thumbnail sketch of your perspective on your space in this translational science discovery space. And I'm going to start at the basic science end with Dr. Lee. Could you give us a brief thumbnail sketch of your work, please?
Collaborating, interacting with physician scientists, including surgical oncologists and medical oncologists and pathologists to maximally translate laboratory findings to clinical application, and also reciprocally to bring clinical information to guide and direct our discovery science.
Sepulveda, would you take us on that next step of translational science?
Hello, everyone. So I was trained first as a molecular biologist, and really my career now spans over 2 decades, progressing from molecular biology to more recent applications in molecular diagnostics, trained as a pathologist, in anatomic pathologist, so I've had a long time practice in diagnostic gastrointestinal pathology in particular. And I've recently joined the George Washington University as the chair of the Department of Pathology and it's thrilling to see a number of great opportunities that we have to grow in this space. I've also transferred my laboratory to the GW Cancer Center, where I've been continuing research that is focused on upper GI cancers, in particular in esophageal cancer, which is funded by the National Cancer Institute, where we have been looking at patient samples as the form of translational research that we do to identify genetic biomarkers that we can use in these patients that have developed Barrett's esophagus. And we have actually published over the past several years a number of studies where we reported a combination of markers that we can detect via doing genetic testing using next generation sequencing and other methods like microarray analysis, PCR, and so on. Some interesting genes have come to the forefront, like a gene that's very well known, CDKN2A, and another gene that is not as well known, but it's quite important in human cancer, which is FHIT, and the third gene that everyone knows, which is TP53. So this combination of markers we have found can really predict the risk of developing cancer in patients who have Barrett's esophagus. And I don't want to take more time right now, but if we have opportunity later on, I can tell you how we're moving forward to bringing this from the bench to the bedside.
Thank you very much. Well, I hope we'll touch on that in a few minutes. Dr. Kaminsky, we know a little bit about your work already, but give us a thumbnail sketch of your portfolio, please.
Sure, no problem. So let me start with a little bit of a story. Back when I exited my residency into a postdoc, my mentor told me, here, write this review article about eye muscles and myasthenia gravis. So there might have been 4 people in the world interested in eye muscles, and now it's maybe 3. But in looking into this, we discovered that these muscles had low levels of intrinsic complement regulators. So every cell has these regulators on it. And these were low, making it likely that they were susceptible to complement injury, which then led to a series of studies on complement treatments and setting up the rationale for phase 2 trials of complement inhibitors, and now ultimately a complement inhibitor that is on the market to treat the disease. But in that way, I developed an interest in really developing novel therapeutics, and we got lucky enough now, which I think is the most interesting work we do, is identifying a protein called survivin that keeps cancer cells alive, long-lived cancer cells, but autoimmune cells are long-lived and difficult to kill, and they also express survivin exclusively. And so just this week, we're talking to a company that might be able to humanize an antibody that I and my colleagues have developed and get it into phase 1, phase 2 trials. And I've been lucky enough to participate in clinical trials over the years. So I'm just an incredibly lucky guy to be able to done all the cool things I did. And most of it relies on my colleagues, for example, Linda Kushner in pharmacology and physiology, and really people across the world that I've worked with.
Thank you. That's really exciting stuff, and covers brings up many questions about this process, about how do you actually do this, to get it there. Really exciting. All right, Dr. Mandy Pratt-Chapman, can you tell us a little bit about yourself?
Absolutely. Thank you. I'm so honored to be here as part of this esteemed panel. I'm an associate professor of medicine and associate center director for patient-centered initiatives and health equity for the GW Cancer Center. And I've had 13 years of funding from the Centers for Disease Control and Prevention, the Patient-Centered Outcomes Research Institute, and the National Institutes of Health. And my work primarily focuses on scaling implementation of cancer control evidence to state departments of health and coalitions, and also advancing health equity through implementation science and healthcare professional training with a focus on implicit bias and organizational change. So, in other words, my work creates some new evidence, but mostly takes evidence that we know and brings it into the community, which might mean patients, or might mean community members, or might mean clinicians or public health practitioners on the ground. I'm actually an alum myself. I earned my PhD in translational health sciences from GW School of Medicine and Health Sciences, and recently was honored to receive the NIH Sexual and Gender Minority Research Investigator Award for this year. And really, I am grateful, and all of my work is really based on the priorities and input of community members, particularly among those historically excluded from clinical practice and research. So I am pleased to be here.
Thank you very much. Okay, exciting stuff too. Dr. Mallinson, can you give us a little thumbnail sketch of your repertoire?
Yes, thank you, Dean Bass. It's an absolute honor to be here this evening. And, it's kind of funny because obviously I'm mostly a clinician. I am a transplant surgeon. I direct the Transplant Institute here at George Washington Hospital. And I'm also the director of the Ron and Joy Paul Kidney Center. And throughout my career, I have been interested primarily in trying to increase access to transplantation to those patients who have the least access, the hardest to transplant patients. The very first project I ever was involved in when I was a medical student at Tulane University some years ago was basically looking at which patients did the best with transplant and which patients had the poorest outcomes. And I was struck to find out that African American patients had the worst outcomes. And throughout my career, I have found that there are two main reasons why these patients have poor outcomes. Number one is they have a problem in getting good access to healthcare. And then number two has to do more with the immune system, immune responses. And that's what my research has centered on. I have some investigator initiated studies going on with the help of philanthropy and the pharmaceutical industry, where we are looking at why is it that patients metabolize medications differently, number one, and then number two, how do these patients' immune systems differ, including their microbiome? So we're looking at blood samples from patients to determine what energizes their T cell and B cell responses, and why are these heightened in certain patients. It's not just minority patients, but we see this more in African American patients than others. And then when you look at the microbiome, which is a hot area of research right now, to look at the bacteria that are in someone's colon and how does that affect immune responses. It's very interesting to me that we are starting to see certain trends based on ethnicity and based on socioeconomics, how your immune responds to different antigens. And this is true both in the serum as well as in the GI tract. And then how do we predict what's going to happen based on this criteria? Industry is very interested in this. One of the newest areas is to look at donor-derived cell-free DNA. So when a patient receives a transplant, we can actually go into the blood and then look at the foreign DNA and how high that concentration is to determine if they are having heightened immune responses. So what I love about the research we are doing now is that it marries two of my favorite areas where I've been involved in since the beginning of my career in order to try to increase access to transplantation for all patients, but particularly minority patients. Thank you.
Thank you for that summary. Really, really informative. Okay, well let's start with a few questions, please, for folks. And I think I'm going to start again, kind of on that basic science side with Dr. Lee. And in your opening comments, you mentioned that you're focusing on a gene that is related to breast cancer. But you also talked a little bit about the importance of having collaboration between clinicians and clinicians and scientists. But first, tell me a little bit. How did you choose breast cancer? So what's your why for studying this particular disease?
Right. Thank you, Dean Bass. So we chose breast cancer first and foremost because it affects so many women and men in this country and worldwide. And within breast cancer, there are several different subtypes. And the one that we particularly focus on is called triple negative breast cancer. The name triple negative because it lacks certain receptor, therefore there's no current targeted therapy. So make it the treatment need much more urgent. And what we found recently is that for many triple negative breast cancer, and this applies to other solid tumor as well, the tumors tend to build a defense line. And the purpose of the defense line is to prevent immune cells from coming in to the tumor and kill the tumor cells. So as you can imagine, this defense line is a major barrier for our immune cells to kill tumors, and also a barrier to the current anti-cancer immunotherapy. And so what we found by collaboration with physician scientists and other researchers is that there is a key instigator of this defense line there that is produced by tumor cells. And the moment you take away this instigator, then the defense line just basically fall apart, and you have a tremendous infiltration of anti-tumor immune cells that come into the tumor and kill tumor cells. And so based on that theory, we have collaborated with clinicians and entrepreneurship and industry partners to design molecules to neutralize this molecule, to break down the tumor defense line.
Okay, terrific. And where are you in that? So your next step for this work would be to get it into the clinical
Thank you. Yeah. So
space. How do you do that?
So we have, with the help from commercialization office at GW, we have submitted a patent, and the GW has secured licensing to a biotech company, and which then use this licensing patented agreement to raise a significant fund to develop therapeutic antibodies, and hopefully in near future we can push this all the way to clinical trials.
Mm-hmm. Yeah, so I think we've all heard this. That's fabulous work, and we've all heard the story that this is often a 10 to 15 to 20-year journey between discovery to actual implementation, and this sort of demonstrates some of that complexity along the way as the many steps involved. And one of I think our overall goals of this panel would be to accelerate that process, and we do that with some of the tools that you've just mentioned, which is partnering with industry sometimes. Let's move on to Dr. Sepulveda, because I think I wonder if you might help us help our group. I'm sure that most of our audience is familiar and fluent in the notion of precision medicine. I wonder if you could just sort of broadly tell us, more precisely what precision medicine means at the pathologic and pathologic and clinical state, and how your work informs that.
Thank you, Dean Bass. That's a fascinating field, and I'm thrilled to speak about it a little bit. So precision medicine, actually, there are several definitions, but in general, it's also known as personalized medicine. And it really involves the use of an individual patient's genomic and other biologic information to make clinical decisions about their treatment. Now, as I mentioned, depending on the context, there are different definitions. One context is in the targeted therapies world, in which we use the right drug for the right person at the right dose. That is the goal, for example, in targeted therapies for cancer. This way, we aim to have better outcomes, reduce toxicities, and improve survivals for our patients. Now, how do we do these targeted therapies? These requires genetic testing of the cancer tissues in order to find specific mutations and other alterations, such as fusions of genes, for example, that are targetable. And these represent the predictive biomarkers of response to therapy. So the partnership between the patient, the clinical provider, the oncologist in particular, and the laboratory, the testing in this application is absolutely crucial because timing is very important. These patients often need results quite rapidly. But also precision medicine can also be used in another context, which I mentioned a little bit about biomarkers in pre-cancer. So biomarkers in cancer prevention can be used for risk stratification during cancer screening and surveillance of patients. And I gave the example of Barrett's esophagus, and just elaborating a little bit more on that, we hope that currently there are over 3 million patients with Barrett's esophagus in the United States. These patients are recommended to have routine and repeat endoscopic procedures in order to exclude progression to dysplasia and cancer. So we reason that using our panel, we may be able to more specifically follow these patients and avoid a lot of unnecessary procedures and endoscopic procedures, but at the same time to intensify that process and or to use treatments such as ablations or chemoprevention in those patients who are indicated. In our process of discovery, we have currently actually submitted our disclosure of invention. So we are waiting for that process. It's, as you mentioned, a very long process. And we are very hopeful that, with a little patience, it will be fruitful. But in terms of precision oncology, I would also like to mention some of our studies and other studies that affect and practices that currently affect patients, in particular in the cancer patient population. So in our laboratory, we have done studies in esophageal, gastric, colorectal, but more fundamentally in esophageal cancer in a more discovery type of approach. We've used what's called a comprehensive next generation cancer gene panel, which tests for hundreds of genes. And in that process, we can identify for individual patients, new genes, new pathways that can potentially become amenable to therapy by drugs that might be currently in the clinical trial setting. So this is very important to do these large panels so that patients might be considered for the early phase clinical trials that are available in large numbers currently. In our study of esophageal cancer using this large panel, which was funded by a private foundation, the Price Foundation, we found, for example, the expected percentage of alterations in the HER2 gene, which can be amplified in esophageal cancer. And so therefore, these patients are candidates for therapy with trastuzumab, much like breast cancer. And this information is very, very useful for their clinical care. This space is fascinating. In the precision oncology now really broadly impacts most tumor types. But among the first were, if some of you might recall, the gastrointestinal stromal tumors with imatinib, CML, of course, breast cancer with the HER2 inhibitors, trastuzumab, one of the first, lung cancer, huge impact, colorectal cancer, melanoma, et cetera, et cetera. And one of the other areas that I have dedicated, given that it's a GI tract cancer is colorectal cancer. Colorectal cancer has a small number of biomarkers that are used for targeted therapies, but most we are performing a number of those in-house, others are sent out. It's very important to really improve the testing of cancers to determine that patients are indeed candidates for targeted therapy. Surprisingly, in the community in the United States, there is still a very large proportion of patients who do not get tested in a timely fashion, and therefore the outcomes are not obviously as good as we all would like.
Yeah, so it really does span that spectrum. We'll hear a little bit about that from Dr. Pratt Chapman, I'm sure. I wanted to go to Dr. Kaminsky now, and you obviously have a vast experience in the whole spectrum of translational science. What I want to ask you about is just the hard work of doing a clinical trial, and particularly doing a clinical trial in a rare disease, as you do. So, two things. Could you tell us a little bit about how do you establish that equipoise when you're developing a new phase 1 trial drug, and how do you present that to patients? How do we persuade patients to be participant in this process? And then, secondly, if you could tell us just a little bit about your rare disease network that you lead now at the National Institutes of Health.
Sure, I'll try to hit on those points. So I think the most critical point that you made is equipoise, right? So it actually starts with the investigators in the field and the clinicians feeling that there's a therapeutic that needs to be evaluated, might be beneficial, and they don't have a bias either way. So I was part of the leadership to evaluate a surgery, thymectomy for myasthenia, which had been around from the 1930s, but never rigorously evaluated. And we collected in that trial 80 investigators across the world, because it's a rare disease. Not all of them need to get this thymectomy. And we actually did a poll, and it was amazing. 50% said, yeah, I think thymectomy works. 30% said, no, it doesn't work. And 20% were honest and said they didn't know. So that set it up. And then we went to find the appropriate patients, explain the situation. And again, going back to that video, trusted them and the investigators to present this in a fashion that they could understand it, appreciate the risks, and have safety mechanisms to assure that if something went wrong, that patient would still be taken care of. So it took years and 4 NIH submissions to get that trial funded, and then another 7 years to go through federal-wide assurances. We had sites in Argentina that needed to be cleared by the State Department. There were just many things over and above just the IRB submission. And nobody hears this, but our investigator in Buenos Aires had to pay off customs people so they could get the sample boxes to send specimens back to the United States, right? So it's a crazy amount of odd things. But again, it came down to organization amongst numerous investigators motivated to figure out an answer, and then to implement the study, continue to work with each of these patients. And it was a 3-year outcome measure. So it takes a tremendous amount of work. And you mentioned the rare disease piece. No center can really identify enough patients to comprehensively study. This is a odd thing, but Barrett's esophagus, that's a great thing to study when you have millions of people in the United States. But with upwards of only 150,000 in the United States, and then there's different subtypes, it's a matter of collecting these patients from excellent centers. That's what this consortium is about. We now are up to 10 sites across the United States that are identifying early onset patients and collecting samples and evaluating each patient rigorously to produce what will be the first real natural history study. And then take those biological samples and go back to people like Dr. Lee and say, okay, can you figure out what the pathophysiology is here? And I'd just like to say one thing about the difficulty really quickly. I think the difficulty is the kind of stuff that Doctors Pratt Chapman and Melanson do. It's one thing to get a drug that's $500,000 a year and nobody can use it. Or there are socioeconomic barriers for patients to even get into a clinical trial or access a new treatment. So that's really hard. I mean, scientists can control everything in the laboratory, clinician scientists control everything in the clinical trial, but once it gets out to implementation, we have zero control.
Yeah, so that's exactly where we were going with this. Next to Dr. Pratt Chapman, in terms of what are the barriers that you face, and what are some of the things that get in the way, and how is your work dealing with that, both in terms of study access as well as implementation to proven therapy access?
Sure. I think that the biggest barriers in terms of, at least we're lucky in the nation's capital in terms of coverage, health insurance coverage. So unlike a lot of areas of the country, which have substantial barriers in non-Medicaid expansion states or the lack of coverage, we have really great coverage in DC for Medicaid. So people are coming through our doors. So that is the first solution, right? Like people need to have access to get into the door. I would say a barrier to that would be when health systems don't take all forms of Medicaid, when healthcare systems don't take a solutions-oriented approach. So, for example, I kind of had been around the system for about 5 to 10 years and was told, you can't do anything about coverage. And back in 2016, I worked with the DC Medicaid office to try to figure out why were patients going to one hospital for surgery and another for radiation and another for chemotherapy. And it was really it was a matter of digging and asking questions and asking the next question, asking the next question, and really developing trust in relationships with government stakeholders, transparency, conducting claims analysis, and focusing on solutions. And we found out that the reason there was so much fragmentation in cancer care, particularly chemotherapy in the district, was because the way that chemotherapy was billed and reimbursed only allowed institutions that had really deep discounts from the federal government through the 340B discount drug program to access chemotherapy. So in 2016, as of May 1st, 2016, DC was permitted by the Centers for Medicaid and Medicare Services to adjust chemotherapy and supportive care drug reimbursement, which blew open the provider network in DC. So sometimes things look good on paper, but we don't know unless we have ongoing conversations across sectors. And we don't know unless we have patient advocates that are identifying problems that come up over and over again in the system, that while it looks really good, it's not working. And so we're lucky in DC that we have great leadership in healthcare finance and Medicaid. They want to do the right thing, but we as healthcare professionals need to also partner and say, hey, this isn't working. Let's figure this out. So I would say, commitment to quality improvement, commitment to transparency and collaboration, strong research infrastructure, and frankly, providers inviting patients to participate in trials would be some of the things that I have seen as barriers to clinical research in DC.
Thank you very much. Last but not least, Dr. Melanson, you mentioned in your opening comments that even some of the discovery science or clinical discovery science you're doing actually is sometimes funded by industry partners as opposed to the NIH or other forms. Can you talk a little bit about how do we put this all together in terms of really having the broad ability to study diseases across many areas?
Yeah, thank you, Dean Bass. And I really appreciate that question because, particularly for people like me who are mostly clinicians, we have some very esteemed members of this panel who spend most of their time as basic scientists and have labs and have a team devoted to basic science. These are the people that get most of the NIH funding, which is appropriate. However, when you want to build up a resume and you want to build up these sorts of studies that could get an NIH grant, it's important to have a lot of preliminary data. So where I have found it very important is to engage my partners in industry to help fund a lot of these preliminary studies. Over the years, we have seen that some of the innovative molecules that have been approved for clinical usage can be used in other ways that are actually more beneficial for some of our patients. This is obviously going to be of interest to the industry, and it's also of interest to us because transplant is one of those areas where most of the medications that we utilize are what's called off-label. So we don't utilize them in exactly the way they were approved originally by the FDA. The industry, the pharmaceutical industry, is always looking for these more innovative ways to utilize their medications. And what I find with some of these most challenging patient populations is that we have to come at a sort of a different vantage point in how to care for them. This is very, very important because, as we have heard stated before, a lot of the clinical trials are not as diverse as they should be. Now, I think we have come a long way in changing this, but a lot of these medicines were approved for use, 10 years ago. So the people that were in those trials back then can be very different from who would make up a trial today with all of the safeguards that we have in place. So for my patients, a lot of the medications that are currently in use, you basically had therapies that were approved in Europe, where you had a very homogeneous population, or even in the United States, where you didn't have a lot of women, a lot of minorities, a lot of African Americans in these trials. And what I've been able to do, and others, we've been able to show, hey, these medicines work a little bit differently in this population. And what they actually respond better to is maybe a lower dose of medicine A and a higher dose of medicine B, and introducing maybe medicine C. So one of the reasons why I'm so interested in looking at microbiomes and looking at the behavior of the immune system is because these tiny little differences, and they're not great differences, but these tiny little differences can have a huge clinical impact. It's interesting when we talk about bench to bedside, but I would also say we go from bedside to bench because it's the patients that teach us so much. So I am interested in, I don't have an NIH grant right now, but I am interested in having one. But I do believe that my partners in the pharmaceutical industry have been very integral to helping us get the sort of clinical data that will lead to a large grant. At least that's what I'm hoping as I write this grant that I'm about to submit.
Yeah, thank you very much. Well, it really does show that, medicine is an art, right? And it does take years and years of clinical judgment and wisdom and observation. So what we do is we observe so carefully and learn continuously from our patients. And when we have the opportunity, as you said, to take that back to our discovery colleagues or our translational colleagues, what a treat that is. And that is actually just one of those core elements, core missions of an academic medical enterprise such as that we have here at the GW School of Medicine, Health Sciences, and our clinical partners. So I want to thank you all very much for this, for participating in this panel. I think it's been really informative. I've learned a lot, and I've really enjoyed it. So thank you very much. And I hope that for those of you who are watching who might be here, should you happen to see any of these distinguished colleagues walking around campus over the next couple of days, please stop them and chat. I'm going to volunteer you all's services for that. So thanks very much, and have a good evening. We'll move on now to our next panel. We have a second one, which is slightly different theme, more focused on innovation and tech and commercialization, sort of the far right end spectrum of translational medicine. So we look forward to joining you shortly. Thank you all.
5, 4, 3, 2, 1.
Well, welcome back, everyone. We've just transitioned to a new group of panelists, all of whom are distinguished alums of our school or our training programs. And this panel will be a little bit different. We won't focus quite so much on the early phase of translational science, the discovery phase, the translation to via clinical trials, but we're going to talk more about basically that innovative process that inspires one to do something new, to figure out what's the gap that we have here. And each of these people has had a remarkable career as innovators and implementers, and even into the commercial space. So it's really important, obviously, that if you really want to have an impact, you got to get it out there, and you got to get it to the patients that need it, and that requires more than just talking about it. So we have three who've really walked the walk here in that regard, and I'm thrilled to have them here with us. Our first speaker is a graduate of the class of 1995, Dr. Laura Obler. She also serves on our board of advisors here at the School of Medicine and Health Sciences. She's an interventional cardiologist at Lenox Hill Cardiovascular Institute. But she also is the CEO and managing partner of MD Concepts, a company that and the founder of a company called Prep, which we're going to hear about in terms of what that does, what her origin was for putting together such an innovative and successful enterprise. We also have Dr. Gail Libovic with us, who is another innovator and extraordinaire, I would say, and entrepreneur as well. She's a graduate of the class of 1986. That's actually the year I completed my residency at GW. And she's the founder and CEO of Silicon Valley Innovations. And she has brought, I believe, 8 different medical technology companies to commercial production. And each has been driven by, I think, what she saw to be a gap in the landscape. And she's going to talk to us a little bit about that process that she uses. Also, we have Dr. Brian Duncan, who was my partner in clinical practice and as academic medicine for, I'm not even going to say how long, but many, many, many years. Also a graduate of the residency program at George Washington University in 1989 in general surgery. He is currently, however, the chief medical officer of Global Boston Scientific Company, which, as most would know, is really one of the world's premier surgical tech, procedural, innovative companies. And that's very fitting for Dr. Duncan, who has always been one of these people who lives just on the cusp of the horizon of clinical innovation and adoption of new technology. So I'm really thrilled to have him with us tonight as well. So that's what I would say about you all, but I want you to say a few words about yourself as well, please, to clarify for us what your repertoire would be. Dr. Obler, could we start with you?
Sure. First of all, thank you so much for having me, Dean Bass. It's such an honor to be here with these incredible panelists. I mean, I am not worthy, but I'm happy to tell you about myself. So I am a cardiologist and always planned on being a cardiologist. Still am a cardiologist. Never planned on doing anything else. Was very happy. Was in a practice. Everything was going well. And then I decided it was time to get pregnant, and I couldn't get pregnant. And it was a time that, I really tried to do everything I could. I really didn't want to go down the infertility route, but I was about to. I got my hands on every book I could read. And one of the books that I read, which was not a medical book, I'd already read the medical books. It was I went to Barnes and Noble and cleared the shelf. And I found a book called How to Get Pregnant Fast. And one of the chapters said, take cough syrup for the 5 days before you ovulate, because cough syrup breaks up the congestion and mucus in the lungs, but it also breaks up other mucus in your body. And the main barrier between a sperm and an egg is cervical mucus. So if you can break that up, the sperm can swim easier. So I decided to do it, decided to try. And it was my 6th month trying. I had an infertility specialist appointment that month. I went there with my husband. We were sitting in the office and the guy said, we're going to start these meds and you need these procedures and you need to get, these tests. And I said, do whatever you have to do. I really want to have a child. And as we were sitting there, the nurse walked in and said, stop everything. You're pregnant. And I said, really? And I said to him, I did something differently this month than the other 5 months. He said, well, did you take cough syrup? And I said, yeah. And he said, well, that's why you're pregnant. And a big light bulb went off in my head. I'm like, this is something I've never heard of. I didn't learn it in medical school. I read it in a random book. How could this be? And that was how I came up with my first company, Preg Prep. And it's a company that helps women get pregnant. And it uses something called N-acetylcysteine, which is a known antioxidant and mucolytic. We use it, with Tylenol overdose. We use it to protect the kidneys and cath. We use it for many different things, and we use it for cystic fibrosis to break up the mucus in the lungs. And there actually, turns out, 8 placebo-controlled randomized trials showing that N-acetylcysteine, if you have infertility problems like PCOS, you're 3 times more likely to get pregnant if you use N-acetylcysteine. So that was really how I started my journey on being an entrepreneur. I brought that to market. I started off by walking down the street, knocking on independent pharmacy doors and saying, will you take 3 kits and 3 bottles, I'll put them in your window. I won't charge you, please. Got a list, got a meeting with CVS, and then ended up in 17,000 stores. And we kind of increased that product line. After that happened, I had a lot of doctors coming to me, as I'm sure the other 2 panelists do as well, saying, I have an idea. Can you help me get it from my head to the shelf? And that's how I started MD Concepts. And I have some other products in MD Concepts, but that's my story in a nutshell.
Yeah. So picking up where the clinical trial ends and just taking it on. It's a really fabulous story. Yeah. Dr. Lebovic, tell us about your pathway and intervention and entrepreneurship.
I'd like to thank you to Dean Bass for having this panel. It's just a pleasure and an honor to be here. What a great story that is, Laura. I didn't know your story before tonight, but that's great. I love it. And I think, it shows that necessity is the mother of invention, as they say. I wish my story was as organized. It's really not. Looking back, it looks as though it was planned, but it really wasn't. When I was young, 14, I had surgery on my knee, and I was so inspired and really in love with my orthopedic surgeon that I decided I wanted to be a doctor. And I had no idea what that meant. But fast forward quite a long time. And while I was at GW, I was inspired after seeing my first mastectomy. I was actually mortified and compelled to change things, because it was a pretty horrifying experience for someone in their 20s to watch that kind with me. And I thought, there's got to be kind of a better way to do this. And initially, I think my pathway and innovation was really mostly clinical, and trying to start different ways of skin preservation. And remember, this was a long time ago. Back in the 80s, skin preservation was kind of a crazy idea. But I kind of aligned with a plastic surgeon who was the former chief of plastic surgery at Stanford when I was doing my residency. And we began to share these types of innovative ideas in the surgical space of how actually to do the surgery differently, and really think about the patient's needs before we did the cancer surgery. And again, this was really the beginning of an entire field that is now called oncoplastic surgery. And it really spans over the last 3 decades of my career, which I love. And during the course of that, I was fortunate enough also to participate in a number of medical device companies. I got a phone call one day from Dr. Tom Fogarty, who many of you will know as kind of the grandfather of medical device development. I got a call from him, and he said, hey, Gail, do you have any ideas in breast? And I said, actually, yeah, I do. And so I started working with him and his engineering team and helped him salvage a company that was kind of um, on a bumpy road. So we came up with a lot of different ideas, and that company got sold. And then we had some other ideas. And so we started another company, and one thing kind of led to another. And we developed a recipe for how to not only innovate new products, identify a clinical need, innovate the products, design them, kind of have a simple but elegant design, figure out the manufacturing, the regulatory pathway, the intellectual property, all the different pieces that go into it. And, you learn something each time. Also learning from other people's mistakes, hopefully. And then what we would do is ultimately do the rollout into the clinical space, the training of the sales team, and then ultimately have the companies acquired. So I've had really a fast and fun ride. It's not over yet. I'm doing yet another company right now. Even though I had no plans whatsoever to do that, the ideas just keep coming. So it's really fun, and I'm thrilled to be here and join you all.
Great, thank you. Yeah. Okay, Dr. Duncan, tell us about your repertoire, your progression through this interesting space.
Well, again, now let me put my thanks in for being part of the panel, and I'm very excited that I get to say Dean Bass. So thank you, Dean Bass, for the invitation. You and I have shared a long journey together, and it's exciting to be together again this evening. I've had a bit of a path, but I think it really, the central theme is leveraging technology to kind of do things better, particularly in the healthcare space. It started very like Gail. I needed orthopedic surgery. I had a shoulder injury in high school. My first exposure to medicine. Nobody in my family had ever been around it. I had exposure to engineering. My brother was an aeronautical engineer. And so I decided I wasn't sure which one of those I wanted to pursue. And so I ended up getting a degree in biomedical engineering. And I liked that very much, but I still was intrigued by the medicine. And so that started my path down medical school and then residency at George Washington University for surgery, which was truly formative, by the way, just a unbelievably fantastic place to launch a surgical career. And while in that surgical training, that was the advent of laparoscopic surgery. So really leveraging technology to transform surgery, take a gallbladder operation that, as you know, used to be quite a long incision under the rib cage and 3 to 5 days in a hospital, and essentially transform that into an outpatient operation. And I saw the power of that. Was just fascinated by how technology could be leveraged to take care of patients better. A laparoscope is a rigid scope that we put in through the abdomen to look inside. I also thought that maybe flexible endoscopy would be needed. We would want to be able to look both from the outside of the intestinal organs, but also from the inside. So then I did advanced training in that regard. And that's kind of been a launching point. Through my surgical career, my interest in using technology to help patients, I became an innovator in technology. And so that's led to being part of forming, I think, 4 companies now, and helping to teach others to use technology in their practice. And then, through my career and more recently, I had the opportunity to join industry full time in the position that you described. I'm the chief medical officer for Boston Scientific's endoscopy division. And, people ask me, well, why did you make the transition out of full-time academic and clinical practice to this? And I think the shortest answer is that the work that my division in the company does alone touches 27 lives a minute. And so that magnifying effect and the ability to impact healthcare. And I tell people, if I do this job right, that's a lot of good that can be done in the world. So that's been my journey, and I just love the fact of how we use innovation and we can leverage technology to do things better in healthcare.
Yeah, thank you very much. Yeah, and I've seen you do that over and over again. It's really exciting. So I wonder, Dr. Obler, could you, you sort of described your pathway to finding commercial partners, and Dr. Lebovic has walked a different way. But Dr. Obler, tell us a little bit about that. How do you launch something of your type? Is it really a door-to-door kind of
Well,
sales
I
pitch?
didn't know what I was doing. I really didn't know. I didn't know what to do. That's how I did launch. I literally did go door to door. I, had a website. I had my 20 little pharmacies in New York City listed, and just kept trying. I mean, I think really the key for me was I just really believed in the product, and I really thought I could help so many women, and it was worth it to me. I mean, it was something that, again, I didn't plan, but it just turned into a passion of mine. And we eventually did raise some money, and as I said, I have other products now and I've raised money, so I haven't gone door to door since that original time. But it was a great experience, and I think that it was a learning process, and it was my path.
Yeah, thank you. Dr. Lebovic, you mentioned Dr. Tom Fogarty, and we surgeons, of course, know him for his work as an incredible innovator, sort of the founder of the discipline of endovascular surgery, which, of course, is now the way all surgery is done. And as a resident, I certainly did plenty of Fogarty catheters for embolectomies. That was sort of the state of the science back 40 years ago. But that really, to me, kind of raises this notion of mentorship and training others, helping others walk this pathway of innovate. Is this a gene, or is this something we can train people and teach people if we create the right systems around them? As we here at GW think about, as we launch very seriously into our innovation venture. What do you think about the role of mentorship and structure on this?
Yeah, I think it's really essential. I think it's very similar to the art and science of medicine, right? We know that we all had a very strong clinical background at GW. I think that was the foundation and the basis for really having solid clinical knowledge and judgment. And then you go to your residency and you build upon that. But stepping outside and innovating and being creative, using that knowledge to advance medicine, like Brian was talking about, is kind of that little piece that we could capture, I think, at GW and put together, as we've discussed in the past, is put together an Institute of Innovation, which I think would be incredibly exciting because there's a lot we can teach and mentor people on. So I think that is an exciting thing. I wound up doing that on the clinical side. I started a nonprofit, the School of Oncoplastic Surgery, and we literally mentor and teach surgeons who are already in practice as well as fellows. We bring them in and we all learn from each other. And that journey never ends. You always keep learning. So I love the idea of mentoring and maybe partnering with industry, which I know many different companies have already done that, try to have institutes of innovation, et cetera. And I think it's essential to keep this going.
Yeah, thank you. Well, Brian, I know that, in your position as head of this, Boston Scientific Global Endoscopy, you must be constantly sort of scanning the horizon for that next great thing, that next technology that you think will be a transformative innovation that will make a difference. So tell me, from big industry's perspective, how do you spot those things? How do you nurture those things? What's the process for that?
Yeah, well, we expend a lot of effort doing just what you described, and we do it on a variety of different levels. So the first thing I tell my colleagues is that that companies like mine, we listen. We go to the meeting, the medical meetings. We read the literature. We're in the procedure rooms watching procedures, and we're using that to gather information about what are the unmet patient needs, where are the physician pain points, and then where do we think that could be useful. and bring that information back to the organization, kind of prioritize it on where we think we can be most impactful, and then use that for for bringing that next great device into the medical community. We also use business development strategies. Big companies would rather invest in small companies to bring their technology inside. It's a much quicker way to do that. It overcomes a lot of hurdles. Big companies are good at making a lot of devices, getting them around the world, assuring that they work properly and function the way they're designed. But sometimes they like to accelerate that kind of pathway from ideation to the real product through small companies. And so we do venture capital work and invest in small companies, and kind of gives us a front seat at the table to see how they develop. And we also purchase other companies when we think that their technology will fit in well with our organization and the other things that we have in our portfolio. So it's a lot of listening. It's a lot of learning. I've been very impressed with my transition from clinical to this, on how well informed, how much these non-physicians know the clinical space and they know the needs, and I think that's a big part. Listen and learn.
Mm-hmm. Great. You know, one thing I heard Dr. Lebovic mention her School of Oncoplastic Surgery. You know, how do you retool, retrain physicians, and clearly in the technology space as well, there's a great need for that. I wonder if you could maybe both speak about what would be the appropriate processes or infrastructure for making sure that these innovations are safely adopted, appropriately adopted, safely adopted. How much does Boston Scientific feel like it owns that space? How much does Dr. Lebovic, when you create something new, do you feel like you're responsible for making sure this is appropriately and safely adopted? Maybe I'll start with you, Dr. Lebovic. That'd be okay?
Sure. I think, it depends obviously on the nature of the device, or as we've heard earlier, the treatments. With devices, there are different classifications of devices, and some are implants, for example, endoscopic tools, some are simple devices. So it really depends on the nature of the device. But one of the things we always want to ensure is that these devices are manufactured appropriately, all the I's are dotted, the T's are crossed, and then the product is trialed on a small basis. So you don't just create a device and sell it to 50 million people or use it on 50 million people. You want to, crawl before you walk and walk before you run. And that's something that our group has really taken to heart and found very valuable, because you learn a lot in that process. As you go, it's just like translational research. As you take your device into the clinical space, you learn more about the patient's needs, how they accept it, but also how the physicians accept and adopt it in their own practices. So that's a really important step. I'd be really interested to hear Brian's take on that as well, like the translation of a device in a small company to a larger company. That's a whole nother step.
Yeah. Brian, you got some thoughts on these two items?
Yeah, it's a great comment, and it's one particularly in procedurally based medicine that we are challenged with more and more. I mean, you and I have worked in this space for some time. It's added that today all knowledge about medicine, all human knowledge about medicine, is doubling about every 18 months, which means that you can't rely on that great training at George Washington University to sustain you through the rest of your career. You need to be prepared to, as you say, retool and retrain multiple times during your career to ensure that you're delivering the best in healthcare. And unfortunately, for practicing clinicians, practicing surgeons, practicing proceduralists, there's no real infrastructure out there to do that. When we were together at Houston Methodist Hospital, we're creating really a world-class institute to do just that, to create an educational home that could be a place where surgeons went to retool and retrain intermittently. And and to leverage the science of education to help do that more efficiently. And that's one thing I would emphasize as well, that, if you think about it, great ideas come to life often through the science of engineering, which is advanced to a great degree. Companies bring those great innovations to the healthcare community using the science of business and the science behind marketing and commercialization. But if we don't leverage the science of education, which is advanced every bit as much as engineering, then education becomes the gating factor that keeps new techniques and new technology from being applied to our patients. And so, industry plays an important role in that. I think the entire medical community plays a role in that. Industry works with experts to design educational programs and then leverage those programs in order to help clinicians adopt new techniques and new technology. And that's a shared mutual interest with a very patient-centered focus. I will say that one of the exciting things that I've been able to do in my transition is take many of the lessons learned from academic surgery and this effort to leverage the science of education to accelerate the use of the safe adoption of technology and bring that over to industry where we can bring resources to bear and really do some interesting work on competency-based training and assessment of performance, and really trying to assure that when you deliver this technology and it's used clinically, that clinician is prepared properly to use it and use it safely.
Yeah, well, thank you very much. I tell you, that's a great way to close this session, in the sense that we started an hour and a half ago with a laboratory studying the microenvironment of a breast cancer cell. And here we are talking about how industry is responsible for retooling, retraining, and bringing the next wave to actual implementation to our programs on a completely different scale. So it's just fabulous. And I really appreciate your insights and sharing your expertise with us tonight. It's been really fun to see the three of you as well. And I hope we'll have a chance to keep these connections going in the future, of course. So I'm going to pass it back over now. I think we're within 45 seconds of our time limit. So I'm going to pass it back to Jackie Wood, who's our director.
Oops.
Hi. So thank you, everybody. I want to say that being part of this panel makes me proud to be a part of the GW family. So thank you to our faculty and to our alumni who participated tonight. For all of our friends who are joining us for reunion weekend, I'll invite you to log off and jump back on at 8 o'clock for our virtual happy hour. There's going to be a link in the chat box to help you get connected. And I know Gail is one of our alumni who are participating tonight. So for everybody else, thank you. We had a couple of questions that came in through the queue that we will answer and use as a follow-up. And then we are also recording tonight, so we'll be sharing that with everybody who participated. But thank you for being part of this. It's a lovely opportunity to connect with everybody virtually, and wish everybody a good night.